I wanted to quickly go over chapter 12. So with this one, we're continuing our work on the nervous system, looking at different sensory things, obviously. So with that, I wanted to remind you first about the sensory neuron. In chapter 11, I told you about the spinal nerves, and that's how we get a lot of input into the central nervous system. And we went over the fact that that nerve will split towards the spinal cord, and you'll have the dorsal root, which will bring in the sensory information, and the ventral root, which will take things out. So that's the motor information. At that time, you have this thing called the dorsal root ganglion. And a ganglion is where we have a collection of cell bodies. Now, if we look at this sensory neuron here, you can see it does not have the classical sort of layout of the cell body, axon, and dendrites that we're used to seeing in those, usually when we teach these sensory neurons. Most of them have the cell body or the neurosoma out sort of towards somewhere along the length of the axon. And these cell bodies will be found in the dorsal root ganglion. Okay, now with these neurons, they will respond to a stimulus of some kind. So that stimulus is collected by a receptor, and in some cases it is the neuron itself. Sometimes it's a specialized cell. We'll go over that, all right? But the stimulus is something that is providing input to to the body that's going to be processed, okay? And it could be all kinds of things, some sort of change in or out of the body. And the receptor then is the thing that is designed or functioning to receive that change, okay? And what its job is to convert that information into some form that the brain can understand. Namely, it has to be converted into an action potential. Now, with this, we have a whole slew of receptors that will detect these things, all right? So everything from mechanical energy receptors to pressure receptors and all kinds of things in between. So thermo, pain, chemo, and photo, all right? Now, of those types of receptors, they're divided into somatic and special. And I think our special senses or our special sense receptors, I think we understand what those are. So taste, smell, hearing, vision, balance. All right, and we know that these are mostly, pretty much, I don't know anywhere else yet, but located in the head, okay? I'm seeing some interesting things in the literature that talk about taste in different places, but that's still really experimental. But, basically, for now, let's say that they're all in the head, okay? Whereas the somatic receptors, these are the things that have that are distributed throughout the body. Okay, and you can see things like temperature and pain and pressure and all these different things. So if they're not a special sense, they're going to be a somatic sense, all right? And when we look at how this works, we can see that the input, the sensory input, most of it is going to be relayed through here, right, through the thalamus. Some of it, though, does not have to be. So things like the olfactory sense, their sense of smell goes right to the brain because the neurons actually penetrate through the cribriform plate and right into the brain, all right? And the other thing is things like body position and movement, that goes directly to the cerebellum, all right? It doesn't need to go up here into the cortex. Although, of course, information about that goes there, but a lot of it just goes right to the cerebellum, all right? Now, how can we tell if something is a stronger a change would be the number of receptors that are stimulated and how frequently they're stimulated, all right? These stimulate the neurons. So the number and the frequency of the action potentials will help us have a perspective on how predominant that change is, all right? Now, when we look at these receptors, I went over that there's several different ones. And here's your ganglion, right? Your dorsal root ganglion. We have what we expect, free nerve endings, and we've got, the cell body, and this is what we're mostly used to seeing, even though this is now a sensory neuron. But we also have the situation where some of these nerve endings will be coated or capsulated. Other ones where the sensory portion is not the neuron at all. We have like a sensory cell that then stimulates a neuron coupled situation here. And in some cases, you can see that this is essentially the sensory cell and the neuron have evolved to be combined, all right? We're sort of thinking of the simplified case being the higher evolutionary path here. Although, I mean, we can't really tell which is, more predominant, but I would assume this would be the more evolved form where we don't need a separate cell for the sensing. Now, so here we have unencapsulated and encapsulated sensory cell neuron pairs. And we'll go over that. Our example there is taste, and the specialized or modified neurons, that would be, our example is smell. When we're talking about the encapsulated, that would be our Pacinian corpuscle, and we'll go over that again. Okay, now one good place to think about all these receptors is the skin. The skin has all kinds of receptors for all kinds of different types of sensations, including temperature and that sort of thing. But also all of the different types of touches. And I was saying in class that if someone is pinching you or tickling you, it's different receptors that perceive those sensations, and they tell the brain that you're being caressed or you're being tickled or there's a bug crawling across your arm. Whatever it is, that sensation even if you can't see it, all right? And it's because they're different types of neurons, and you can sort of see some of them are going to be modified and encapsulated, some will not be, all right? And the type of information they're sending to the brain is a little bit different. So here we have, and you'll also notice that some of them are located in different places. So light touch is going to be closer to the epidermis, whereas a deep pressure, like the Pacinian corpuscle, is going to be further away, right? Others are going to be located along the hair. Some are going to have free nerve endings penetrating right up into the epidermis. We had talked about the Merkel discs as well, that they sort of will reside here. This is sort of, that's going to be a two-cell system. If you can remember, we had the Merkel's discs in the epidermis, and there would be an underlying neuron. Whereas here we have, modified ending, right, encapsulated. Okay, so slightly different situations, different types of neurons and receptors and pairs. And this is how we get such a variety of different types of sensations by the skin. And notice with the temperature, we have cool receptors and warm receptors, and we're going to play with those a little bit in lab. Now, what would be the purpose of having an encapsulated system? And the example here is the Pacinian corpuscle, and this is the one where we sense deep pressure, so way down in the dermis. Now, the thing to notice here is that when you feel it, right, you've got an increase in action potentials, but with time, you can see the sensory part of this neuron will relax and straighten out. So even if that pressure is still there, you'll get much fewer stimulations of that neuron. And you know that things like deep pressure, you stop thinking about them, right? So if you're sitting on a chair or standing on your feet, you know for a few minutes you're very conscious of the fact that you're sitting in the chair or you're standing on your feet, okay? And then you forget about it, and you'll start thinking about standing on your feet if you do it long enough, and you start initiating pain receptors, because those are different, okay? Those are ones that don't, in this case, adapt, and that's the purpose of this slide here, is to talk about sensory adaptation. So certain types of things we will adapt to, and why do we do that? Well, so that the brain can move on to other things, okay? So things that adapt are things that, pretty much don't necessarily mean death if you don't pay attention to them, okay? Whereas other things that do not adapt would be things for pain and the positioning of your joints and muscles. You have all kinds of special sensors in your body that just tell you about body position, okay? We don't adapt there either, because it wouldn't be good if you forgot about that, the fact that you're standing or walking up a flight of stairs. You don't want to forget that. So these are the really important things that, again, could, influence the well-being of your body. Whereas light touch, a light touch really doesn't tend to be something that could be harmful. So you eventually adapt to that, for example, or the pressure from the chair that you're sitting on. And I had mentioned, at least in one of my classes, that this is how pickpockets take advantage of the fact that a lot of usually men, keep their wallet in their back pocket. And that's because eventually that deep pressure sense is going to, you'll forget about it. And then what a pickpocket will do will distract you, like tap you on your shoulder at the same time as pulling out your wallet. And you'll eventually notice it, right? Because you'll have lost that pressure and your brain will say there's no more pressure. But that's why they distract you a little bit when they pull the wallet out. And by the time you notice it, you're probably too late, okay? Then we have those deep pain receptors. Like I said, pain is not something that you forget about, all right? So we have, these are unencapsulated nerve endings, and fast pain, you know the difference. Fast pain versus a slow pain. Fast is acute, sharp, happens really quickly, and you try to avoid it right away. Slow pain, it comes on and it just stays there. And this is the referred pain that it stimulates here. And in order to remember why do we get referred pain, which is basically that we receive pain originating someplace else. Why do we have that? Well, remember, a nerve has neurons that are running from different places in the body. Some of them are going to be coated with myelin, some of them will not, right? So we can have some cross talk, or not talk, but you could have some influence on those ones that are not myelinated. And also, since you've got these nerves carrying information from different places in the body, sometimes the brain gets a little confused, right? It sort of crosses information. And basically remember that those nerves carry information from different places in the body. And the classic example that most of us are familiar with is when men have a heart attack, they start to feel the pain down their arms instead of in their heart muscle. Well, they will feel it there too, clearly, eventually. But very often this aching of the arms will occur. It doesn't happen the same way with women. Women have other feelings, sensations such as nausea and that kind of thing. Very often women who are having a heart attack think that they just got hit by a flu bug. They'll wake up and they'll feel nauseous and sweaty. So it's a different type of referred pain, but it's also referred. But this thing with the arms, this is typically a male heart attack, all right? And you can see other examples here, all right? But just remember, referred pain is when you're feeling pain someplace else than where you're actually suffering that pain, all right? Now, getting into our special senses, the first one we're doing is taste. That, of course, those sensors are located on the tongue. And here we have that situation where we have a receptor neuron pair, okay? So here we have these special sensing cells. We have 5 different types, and these are chemoreceptors. So they're actually sensing a chemical, all right? They're not sensing sweet, they're sensing glucose. And salty, they're sensing sodium. For sour, they're sensing acid, okay? So the hydrogen ions. Bitter is it's a structure that stimulates that. There's a few things that have that structure. Umami is the one that is a new one. We're still trying to understand this one. But this one is a savory flavor. It's the thing that tells our brain that this is tasting good. And the locations here I'm covering because that's sort of what your text is saying. But if you look into the internet, I don't know the internet, not the internet, that's not the right word. If you're looking into where these senses are found in the literature, that's the word I was trying to use there, the literature, there's sort of a difference of opinion here. And some people say that it's not even true, could be, and it's because it varies so much between individuals. But classically, when we think of where is sweet, and this is what you need to know for lab, like where is sweet, it's mostly on the tip of the tongue, salty, the tip and the sides, sour on the sides, but in a different place than salty. Bitter, mostly at the back. And a lot of medicines have that bitter flavor because they have that molecule in there that stimulates those cells. Now, these tastants, or these things that we taste, these flavors, are called tastants. They have to be dissolved. And that's why, well, that's not why, but the benefit of having most of your saliva be water. They dissolve these chemicals, it dissolves these chemicals, and in so doing, you can get through the taste hairs here and stimulate the taste cell. All right. And you can see in the taste bud that there's also some support cells in here, but really what you need to know is that the taste receptor is linked to the neuron. Okay. Okay. Now, smell is a little different. And you can see here, remember that those neurons that sense smell, they go right to the brain, all right? And here we have the modified neuron sort of structure. We don't have a separate sensing cell. It's the neuron itself that's sensing this. You can see that they're located right here at the top of the nose. This is the cribriform plate, all right? There's little holes there, and these neurons go right through there to stimulate the brain directly, right? So it's right into the olfactory bulb. These are those modified neurons. And look, we have over 1,000 of these. So we have over 1,000 completely different sensory neurons. They don't sense the same chemicals compared to only 5 flavors or tastes. We have over 1,000 odorants that can be detected. Dogs have many, many more. For some reason, the number 10,000 is in my head. I don't know why, but that is very different. So you can tell this for a sense of smell. While our sense of smell is much more developed than our sense of taste, the dog's is even more developed, all right? Now, this correlation between taste and smell, you know, when you have a stuffy nose, you cannot taste food half as well as when you don't. And these are linked. And this is because a lot of the things that we taste are also sensed by the nose because they're chemicals. They have to be dissolved in water, right? Same thing. So the mucousy, watery mix in your nose. And those chewed foods, when you chew food, it releases these little particles into the air that can be inhaled. Other things come up through the back of the throat, right? Because remember, the nose and the mouth are connected at the back with the pharynx. So there's a space there. So when food is released, those chemicals are released. When food is chewed, those chemicals are released and they can stimulate your olfactory receptors just as well, okay? Now, hearing, I should have hearing and balance really is the title on this slide, but we're going to look at this for the ear, and with the ear we have to sort of go over what these structures are. So on the outer ear, that is responsible for collecting sound waves. It ends at the tympanic membrane. Here what's happening is it's transferred to the middle ear, and the job there is to amplify the sound so that they become more predominant. And then the inner ear, the job there is to convert that sound information into action potentials. All right, so that for sound specifically, that's going to happen in the cochlea. All right, and I love this slide where you can see the bones of the inner ear attached to the tympanic membrane. I just think this is really cool. Anyhow, now the nose is connected to the ear through, well, not the nose, the back of the throat, so that the whole area there is connected to the inner ear using the eustachian or auditory tube. Okay, those two are the same thing. There's just two names for the same tube. So eustachian or auditory. I think your book is using the term auditory, and you might have learned it in past as the eustachian tube. But anyways, the point here is you can see the connection here to the pharynx, right? So there's this hole there. It's not covered, so mucus can easily get in here, especially when you have a cold. And the reason why young children get inner ear infections much more easily than adults is because if you see this eustachian tube, I learned eustachian, so I tend to use that term more often. As you can see, this eustachian tube is almost horizontal, so it's really easy for junk to get stuck in there. Whereas in an adult, there's more of an angle so that it will drain out. As you know, it's not perfect, right? We've all had that stuffed ear when we have a cold, stuffed ear feeling. But it's worse for children, and it festers in there and the bacteria start growing and they get an infection, and it's just a disaster. But yeah, that brings back memories. Anyhow, this is why this happens. All right. Now, when we're talking about the inner ear, it's not just hearing. We also have balance, and the sense of balance is sort of covered by two parts of the inner ear. So, and they are coupled together by a term called the vestibular apparatus. And that vestibular apparatus includes the vestibule, which has the auricle and the saccule, and these govern balance and acceleration. And the semicircular canals, there's this swelling here called the ampulla, and that's where the sensors are for rotational movement. So those together are going to help you with your balance. All right. Now, cochlea, obviously, that's the one that's left. This is where we have our hearing. Now, in order to really understand how these work, all of these sensations, rotational movement, acceleration, balance, and hearing, all of those use the sensory cell system connected to a neuron, and the sensory cells for all of these are called hair cells. And you can see they're diagrammed here, literally have little hairs on them. And they give a certain amount of action potentials as a baseline, and that would be when the hairs are erect. Now, when the hairs bend, they will be giving either more or less neurotransmitter, depending on what kind of hair cell we're at, what we're sensing. Yeah, for hearing, it's mostly this situation, but the rotational movement and the balance, that one, we have a situation where we could have more or less so that your head knows this way or that way, right? Forward or back. But the point is that if they're bent in one direction, they'll stimulate action potentials. Bent the other way, it's possible for them to have less. All right. Now, how these cells are bent or how these hairs are bent will vary between the different situations. Okay. Now, let's go over hearing first. And I recommend watching this video again. This is quite involved, so there's a lot of steps involved here. So let's walk through it. All right. So first of all, the sound waves are coming in. They're going to hit that tympanic membrane or the eardrum. And that is then going to move, and in so doing, it's going to move those three bones, right? The malleus incus and the stapes. The stapes then is connected to this round window. Sorry, the oval window. I was just distracted there. My husband opened my door here. Not sure what he wants, but anyways, it vibrates or moves the oval window. And there's a fluid in this cochlea, all right? There's two different types of fluids. But basically, this fluid is going to start to move, right? We're going to get like waves in this fluid, all right? And this part of the space, this part of the cochlea is called the vestibular canal. Now, you can see that this sort of goes around to this side, okay? Now this, it really is connected, all right? But we have, you can see there's a term for the place where we move into the other part, where we're moving sort of back here. So that's called the heliotrema. You don't need to know that for this course, but you'll see it, you know, and in here you have the scala tympani. This is the other side of this, whereas here on this side is the scala vestibuli, all right? But really one continuous sort of chamber. So now why do we have this? Well, if you have very, very loud sounds, this is going to go through this fluid all the way around, and we have to relieve the pressure. Okay, so there's a reason here. So this is going to be release that pressure here at the round window here, over here, okay? So that the sounds don't reverberate and go back and stimulate again, all right? So that would be a bad thing, all right? So this is sort of why we have this setup. Now, how do we hear things? So what's going to happen here is if we have high frequency sounds, a membrane is going to be vibrated closer to this oval window, okay? And this, this membrane here is called the vestibular, all right? So here we have it here. This vestibular membrane is going to vibrate closer to the oval window, all right? You can see it's going to sort of have an effect. So closer to the end of the cochlea that is by the round window or the oval window, but it really is closer to the oval window. Now, if we have a more low frequency sound, it's going to flex that membrane, okay, that vestibular membrane further away from that oval window, Okay, so this is how the brain can tell a high pitch from a low pitch sound. All right, now we still haven't told you how the hair cells are bent. Okay, all right, so let's get into that. All right, so if you're following along with my notes on point 6 on page 7, all right, now, so the lower frequency sounds are going to vibrate the vestibular membrane further away, higher frequency sounds closer together. Now, what's going to happen now is that we have fluid within the organ of Corti, which is where we actually are now going to sense the sound, all right? This organ of Corti here in the middle. And what's going to happen is that there's fluid in there again, all right? So there's another set of fluid in here. This is called the endolymph, all right? And you'll see that, yeah, there's another membrane here, okay? So let's move on to the next slide, right? Because this is where I'm going to follow along with my notes, okay? So we're at point 10 now. So the vibration of the vestibular membrane then is going to vibrate that endolymph inside the cochlear duct. Now, this fluid, when it's vibrating then, is going to vibrate this fluid now. This is now called the tympanic canal, all right? And what it's going to do then is it's going to start vibrating another membrane as well, all right? So the other membrane that it starts to vibrate is called the basilar membrane. Okay, so the basilar membrane begins to vibrate because of this movement of this fluid here. Okay, now when that happens now, those hair cells are sitting on this basilar membrane. All right, so if we look down here, we can see that the hair cells are situated. This is the basilar membrane, and these hair cells are sitting here. And their hairs are embedded, right? Bedded into a third membrane called the tectorial membrane, all right? So as these hair cells, and then we'll look here, as these hair cells are bent because the cells are going to be moving up and down on the basilar membrane, that is going to bend the hairs and that's going to send information to the brain, all right? Now, where that happens will depend on whether that is a low-pitched or a high-pitched sound. All right, so the action potentials that are generated by the bending of these hairs on these cells are going to go through the nerve. It's called the vestibulocochlear nerve, and that is going to get to the brain. All right. Okay. Now, I would recommend watching that video because that will help a lot, all right? There's a lot of little things going on, right? We have the tympanic membrane, we have the vestibular membrane, the tectorial membrane, the basilar membrane. So they're trying to keep that all in perspective, right? And we have the different chambers. We have the endolymph in the middle, right? And the perilymph on the outside. So to try and keep that all straight, okay? Okay. Now, rotational movement. Again, we have hair cells, but in this case, the hair cells are not banging up against the tectorial membrane when a basilar membrane is being bent or fluxing. What's happening here for rotational movement, and again, remember this is now in the ampullae of these semicircular canals, we have hair cells that are sitting on a sort of a nice little raised thing here. We call this together, we've got this sort of support, and we've got the hair cell, and we've got the bundle. This is sort of called the cristae. And you can see that these hairs are embedded into sort of a gel called a cupula. All right, so here's the ampulla. Inside there we have this cupula that is sitting on top of these hair cells. Now, what I want you to imagine here is, think of the ocean and the swaying of corals in the ocean, right? So when the current flows one way, the corals bend that way, and then it flows back the other way, the corals bend the other way. All right, so this is what's happening with this cupula. So it's in this fluid that's sitting in this ampulla, and this cupula is sort of swaying in the fluid, and it's when you rotate your head, the fluid is going to sort of move more quickly, and then the cupula is going to follow it, And then if you move your head back again, the fluid will move, and then slowly the cupula will follow it the other way. And when it does that, it is going to bend the hairs. All right. So this, like you can see here, the rotational movement causes the fluid movement sort of in the opposite direction in the semicircular canal. And this fluid then is going to push against the cupula, it's going to bend it, and it's going to activate those cells. So in that way, whether the hairs are moving one way or the other way, your head will process that to know that it's, you know, how you're shaking your head, all right? Okay. Now with head position acceleration, now remember now we're going to the vestibule, all right? And in there we have a little portion called the auricle, and we have another one called the saccule. And again, we have hair cells that are connected with neurons. And again, these hairs are embedded, okay, in a gel, all right? But in this case, what we have now here, what we're sensing is acceleration and head position, all right? So what these this gel is going to respond to is gravity, all right? And to do this, there are these crystals, all right? So these crystals that are sitting on this gel, those crystals are called otoliths. They're heavy and they're pulled by gravity, all right? So if you think about it with the head position, right? What's going to happen if you move your head forward, the gel is going to move, those otoliths are kind of heavy, you're going to pull that gel forward, and it's going to signal the brain in one way when the hairs bend that direction, and if you put your head back, you can see that the hairs are going to bend the other way. Now, think about the same thing is going to happen with acceleration, all right? So you'll be sitting, say you're in the car, and then the car will speed up, and what will happen is that that gel is going to get sort of pushed back. I'm sure you've had stuff in your car that went flying one direction or the other direction, depending on whether you're going forward or back. This gel is going to do the same thing, and the otoliths are going to shift that gel so that your brain knows how, in what direction you're accelerating forward or backwards, okay? All right, and that's how those cells are going to be stimulated. Okay, I'm just going to look at here, see if there's something else I should tell you. Okay, nope, that's okay. It's fine, good enough. All right. Now, vision. All right, so the path of light through the eye. Now here, and this is a good video, watch it again, all right? So what's happening here is that light, when it comes to the eye, first thing it's going to pass through, all right, is the cornea. The cornea is a clear part of the outer portion of the eye, which is actually called the sclera, right? So it's clear in the front. The light passes through there, and then it's going to go into this fluid-filled space. It's called the aqueous humor. And then it's going to go through the pupil, all right? The pupil is surrounded by the iris. That's the colored part, okay, of your eye. The iris is actually a muscle, which we'll go over, circular muscle. And the pupil is actually just a space, all right? And that space, behind that space, we have our lens, okay? So then the light goes through the lens. Then after it goes through the lens, it goes through a fluid called the vitreous humor, and I'm going to go over that in a sec. And then it's going to hit the back of the eye where we have the retina and our photoreceptor cells. And I'm going to go over that a little bit more carefully. Now, I just want to mention something about these two humors, the aqueous humor and the vitreous humor. Now, the vitreous humor is in the sac, right, of the eye. This is not circulated, so whatever vitreous humor you have is what you've got. And this is why you start to see floaties sometimes. We call them little floaty things, and that's because there's a buildup of crystals or whatever in this space, and you can't get rid of it. You know, you have it, and sometimes you'll see it because it will occlude the light coming through your eye. The aqueous humor, on the other hand, is circulated. It's out here, it circulates, and there's a drainage system here that will allow it to go back into the bloodstream, all right, on this side of the lens, all right, so it actually circulates here, and the vitreous humor is behind the lens, okay, but that one does not circulate. Now, in order to have vision, we have to do two things. We have to focus, right, and we also have to process the image. Now, focusing, the way that works is done by the lens. Okay, so the lens is what's going to focus the light on the retina. And this act of focusing, all right, is involving the bending of the light by the cornea and the lens, all right? Because if you pass light into water, it's going to bend. It's going to happen the same thing here. This is fluid-filled space. It's not air. It will bend a little bit here, but your lens, the way we deal with this is this process called accommodation. And this is actually the changing of the curvature of the lens for near or far objects. And this is done by a muscle that holds onto the lens. Now, you can see here, it can be a nice fat sort of cornea or a tension or loss of the tension on the cornea is going to change the shape, all right? And the muscle here is called the ciliary muscle. So when the ciliary muscle is contracted, what happens is that the lens has one shape, and when the muscle is relaxed, it has another shape, okay? So when it's relaxed, these fibers get very taut, and the lens stretches. When the muscle is contracted, what will happen is that these will get slack. And you can see that it will focus depend accordingly. Now, as we get older, we end up having to use reading glasses, which is what you're seeing on my head most of the time, all right, or down my nose, and I look at you over them. Now, this is because as you get older, all right, when you get to about my age, not that I'm old yet, but when you hit my age, around, and I'll not tell you, even though I just had my birthday, what will happen is that muscle won't be as strong. All right, so it's not able to accommodate to focus as well, and then we need some lenses to help us with that. Now, these lenses that I have are different than glasses that you guys probably have. This is for a different reason. Okay, so I'm going to talk about that in a sec. All right, now the amount of light that comes into your eye is regulated not by the lens, but it's regulated by the iris. And literally the iris is two layers of smooth muscle, right? So we've got radial and longitudinal. Radial and circular in this case, circular and radial. These will contract differently depending on whether it's open or closed. So if the pupil is dilating, the radial muscles are going to contract. It's going to open up the pupil. So dilation means that the pupil is open. When the pupil constricts, what's going to happen here is that the circular muscles are going to get tighter, okay? And that will regulate the amount of light entering your eyes. So when it's dark, this pupil is really open, okay? That hole is really open. And when it's bright, it closes down. Now, when you see that red eye in a photo, this is because the iris is open too far for the flash, and that red is actually a reflection of the blood vessel, the blood from the blood vessels in the back of your eye, and that's what's making that red color, all right? Okay, now, for you guys, you probably don't need reading glasses like I do because my ciliary muscles are getting weaker. What you guys have, the glasses that you have, is due to the shape of your eyeballs. Okay, that's different. We're not talking about this muscle anymore. Okay, so a normal eye is called emmetropic, all right? That's someone who doesn't need glasses. And what will happen for them is that light can easily get through and focus exactly on the retina, right? Without any help. But if you're someone who is nearsighted, you're myopic, what will happen here is that your eyeball is kind of squished down. So you've got an elongated eye. And what's happening is that the image is focusing too early, right? So before it hits the retina. And the way that's corrected is by using a concave lens. So that will move that that focal point back towards the retina. Now, for someone who's farsighted, I'm a little bit farsighted. So I also have glasses for driving, especially at night. Seems to bother me more at night. But if you're a farsighted person, your glasses, right, they're going to bring the focal point closer, because what's happening to you is that the image is focusing behind the eye. Why? Because the eyeball is a little squished vertically, all right? And this would be hyperoptic, all right? So what's happening here? And in order to correct for that, we have a different shaped lens. We have a convex lens, all right? So understand the difference between reading glasses for someone like me or your parents, whoever, versus, you know, having nearsighted or farsighted vision, all right? So that has to do with the shape of the eye, whereas reading glasses, that has to do with the strength of this muscle and its ability to accommodate for close or distant objects, all right? It's a little bit different. Okay. Now, when the light gets to the retina, what happens? Well, it's kind of a weird thing because you would think that your photoreceptors are right there to receive the light and it just goes to the brain, but that's not the way it works. So there's actually layers of cells here, and the photoreceptor cells are actually the furthest away from the surface. So you've got your rods and your cones. These are your photoreceptors. We're going to go over those in a sec. All right, so the outermost layer of the retina is actually some pigmented cells. You'll see if you get to ever dissect an eyeball, you'll see that there's this layer of black cells here. And they prevent the light from scattering, all right, and absorb excess light. And then you have your photoreceptors. So the light is going to go all the way through this thick layer of cells. Back here, here are your photoreceptor cells. Now you have a lot more photoreceptor cells than you have neurons. So what ends up happening here is that there's a layer of cells called the bipolar cells. And this is where you have a situation where you have convergence. So you have many cells sending information to one cell. So we lose some photo information. It goes to the bipolar cells. And then at the innermost, then we have our actual neural ganglion cells, all right? They're here, and then those ones, all of the axons from them form the optic nerve, which then exits the eye. And this is the blind spot, right? So we literally have this weird situation where we have a blind spot because these neurons leave right through the retina. Okay, so it's kind of backwards, all right? So the light goes to the back, and then the sensory information goes back out towards the vitreous humor, and then through this axon, okay? So we have this situation, we have convergence of the information, all right? But we're also amplifying signals. We have this, you know, summation here. So let's go through this. So the innermost is the ganglion, that becomes the optic nerves. So the photoreceptors, they're going to respond to the light, right? They have pigments in them, all right? These pigments are proteins. They change shape depending on whether they're stimulated or not. When with this, we have about 120 million rods. Those are going to detect just light, but not color. And we have about 6 million cones, but we only have 1 million ganglia, all right, back here. So what we're having is convergence of the signal, but an amplification of that because we're losing signals. We have convergence and summation that's happening here. Okay, hopefully that makes some sense, all right? Now, what are the difference between these cells? I already sort of talked about this. Now, we have basically 3 types of color receptor cells and another type of cell that just detects light and dark, all right? But in each case, there's a photopigment that gets stimulated, all right? So when we look at our rods, the photopigment here is called rhodopsin, and it's much more sensitive to light than the photopigment in the cones, all right? They're a little slow though, all right? So, you know, this is what we rely on the rods for vision in dim light, all right? Primarily depends on that because there's just not enough light to stimulate the cones. So the rods don't give us color vision, but we pretty much see black and white at night in dim light, okay? And most of them are not found right at the center of the retina. They're most sort of back around the edges a little bit more. Not that we don't have them in the center, but there's less. So we have most edges around the edges of most rods around the edges of the retina. Now, when it comes to color, we see 3 colors, red, green, and blue. There's other animals that see more colors than we do, all right? But the variety and the number of these ones stimulated give us all of our hue information. So our shades of red, or shades of blue, pink, and orange, and all of this stuff. So we don't have like an orange pigment, but we see a combination of red, green, and blue. And these cones where we detect color, so C, cone, color, right? They require more light to be activated, so we don't see color at night as well. but they're really responsible for the sharpness of our vision. This is why during the daylight time, our vision is a lot sharper, okay? That's probably why my farsightedness kind of bothers me more at night than it does during the day, because things are just not as, sharp. But most of our cones are towards the middle of the retina, okay? Now, when we look at this, when we look at the retina, we have sort of, different things here to look at. I've mentioned the optic disc. This is our blind spot. This is where we don't see anything. All right. Now, the way we compensate for that is that the blind spots in the two eyes are slightly different, and our eyes are constantly moving. We'll talk about this in lab, but it sort of shifts all the time. So your brain sort of compensates for that blind spot by constantly moving. It's not possible to keep your eyes still unless you're dead. Okay. Now, we have this area where we have a concentration of our photoreceptors, and that's the macula or the macular retia. And most of that is going to be photoreceptors. And right in the middle, there's this little groove called the fovea. And that is where we have the highest concentration of our photoreceptors. Okay. All right. Now, things that can go wrong. Well, there's retinitis pigmentosa. This is a hereditary condition. And what this involves is the progressive degeneration of the rods, all right? And in that case, you're sort of losing your outer vision and your vision in dim conditions. And so this is sort of what happens. And you end up having to, eventually you lose all of your vision, but you lose it from the outside in. And this is progressive. And there's still a lot of theories on that, but basically, I used to put the paper up for other courses, but basically what's happening is that the rods are important for the health of the cones. There's something interesting happening there. They communicate with each other. So once you start losing your cones, eventually, your rods, you'll eventually lose your cones, but the rods go first. Now, that's different than macular degeneration. So remember, the macula is where we have most of our photoreceptors. So here, the degeneration is happening in the center. So it gets really hard to read and look at faces, okay? So you're sort of losing it from the middle outward, okay? Cataracts, that's when the proteins in the lens lose their alignment. So it's a clouding of the lens, and the way they treat that, basically, and that happens to a lot of older people, but it happens to younger people in other countries where it's difficult to treat this. So a lot of people go blind because of this. There's no reason to, because what happens is the lens is replaced with an artificial lens, and that's how that's dealt with. Then we have colorblindness, primarily red-green. So if you're looking at this image and you do not see the 12 that's there, then you are red-green colorblind, all right? So what's happening there is that you're just not making that pigment, okay? It's very common. You don't have those cones for the color that you're missing, okay? Then the last one is glaucoma. And this is caused by an increased pressure in the eye, all right? So what's happening here is that the aqueous humor actually builds up and the pressure builds, and eventually blindness will occur. And it's kind of a weird sort of effect. I mean, I don't know how they get these pictures, but I guess they listen to a description of what people see who are suffering from glaucoma. But you can sort of see the image is a little distorted and we're losing it on the outside. But eventually it results in death of the photoreceptor cells. Okay. And that's it. All right. So hopefully that has helped. And we'll go on with chapter 13.